Hello everyone, it's time for another warm-up. Let's get going. First one here, which cardiac defect is suggested by each of the following types of murmur? So first one we have here is a pansystolic or holosystolic murmur, heard best at the apex and then often radiates to the left axilla. Think of mitral regurgitation. Crescendo decrescendo systolic murmur, heard best in the second and third right intercostal space close to the sternum. Think of aortic stenosis. Late systolic murmur, usually preceded by a mid-systolic click. That's going to be mitral prolapse. Early diastolic decrescendo murmur, heard best along the left side of the sternum. This is going to be a pulmonic regurgitation, or it could be aortic regurgitation. Late diastolic decrescendo murmur, heard best along the left side of the sternum. So this would be a tricuspid stenosis. Next, which heart defect is associated with each of the following disorders? So first we have chromosome 22q11 deletion. So this is associated with truncus arteriosus and tetralogy of Fallot. Keep in mind that the question is asking for heart defects, but definitely also know that that 22q11 deletion is well known to be associated with DiGeorge syndrome. Down syndrome, so that's going to be an endocardial cushion defect. Next we have congenital rubella. So this is either a PDA or pulmonary artery stenosis. Turner syndrome, so this is going to be coarctation of the aorta, also a bicuspid aortic valve. And then we have Marfan syndrome, so think of aortic insufficiency. All right, that brings us to the end of the warm-up. Let's get to that lecture now.
We've discussed how the immune system can be divided into innate immune system, which is primitive and nonspecific, and the more specific adaptive immune system. Well, you can also divide the immune system between cell-mediated immunity and humoral immunity. Cell-mediated immunity is basically T cells or macrophages or NK cells, cells that attack pathogens and eliminate them. And then there's humoral immunity, which means antibodies. The word humoral has to do with body fluids, like back in the old days when they thought disease was caused by evil humors, and they would treat by bloodletting or diuretics or making the patient sweat, to get those bad fluids or humors out of the body. So humoral immunity means that this is the acellular part of the immune system that's floating around in the body fluids, specifically antibodies or immunoglobulins. Now, where do antibodies come from? Well, when a mommy antibody and a daddy antibody love each other very much, antibodies come from B lymphocytes. So let's talk about B cells and antibodies. Let's start with some B cell basics, like T cells. B cell precursors originate in the bone marrow. But when T cell precursors leave the marrow and mature in the thymus, the B cell mature in the bone marrow. So just like we said T in T cell stands for thymus, let the B in the B cells stand for bone marrow. And then once these B cells are mature, they migrate to the secondary immune tissue, like the lymph nodes and the spleen and the molt, where they hang out and wait to be activated. Now, what two cytokines did we say Th2 cells produce that stimulate B cell proliferation? IL-4 and IL-5. But when B cells encounter antigen and are activated, they differentiate into plasma cells to produce antibodies. Or the B cell could become a memory B cell, which goes dormant and waits around in the case that same pathogen reappears. But as you age, the number of memory B cells gradually decreases, and that's why some immunizations can wear off as you get older. Like T cells, B cells have very specific surface markers, and there are 5 surface markers that I think you should know. There's CD19, CD20, CD21, IgM, and IgD. We'll talk about the specific functions of different immunoglobulins in a minute. But remember that IgM and IgD are found on the surface of B cells, in addition to CD19, 20, and 21. We also talked about how B cells can act as antigen-presenting cells because they present antigen to the Th2 cells, and then that Th2 cell can start producing IL-4 and IL-5. So if they are presenting antigen to the Th2 cells, that means the B cells must have MHC2 on their surfaces. And they also must have a B7 protein to help activate the T cell. That B7 protein is important for providing the co-stimulatory signal that stimulates the T cell. And then we mentioned that B cells have a CD40 surface marker that is part of the T cell co-stimulatory signal to activate the B cell. So there are signals for the B cells to activate T cells and signals for the T cells to activate B cells. While we're here, let's talk about the surface markers for other cell types. So these are the surface markers we just covered in B cells: CD19, CD20, CD21, IgM, IgD, MHC class II, a B7 protein, and CD40. Then, T cells have CD3 and a T cell receptor, and also CD28, which is what binds to the B7 protein on the antigen-presenting cell. Cytotoxic T cells also have CD8 surface markers, in addition to many others, and that CD8 is what helps it bind to MHC1. And helper T cells have CD4, plus they have a CD40 ligand for activating B cells. The B cell presents the antigen to helper T cells and says, "Check out what antigen I found. What do you want me to do with it?" And the activated helper T cell can tell the B cell to start making antibodies. Natural killer cells have CD56, which is the unique marker for NK cells. And they also have CD16, which is what binds the antibodies that coat infected cells and initiate antibody-dependent cell-mediated cytotoxicity. Let's also look at the surface markers for macrophages. We'll talk about macrophages in another video, but macrophages have CD14 marker. And then macrophages are another type of antigen-presenting cells, so they have MHC2 and B7 protein. And they have a CD40 marker, just like the B cells, which the Th1 cells use to make them more sensitive to interferon gamma. Again, we'll talk about macrophages separately. And then, just as a reminder, all nucleated cells have MHC1. Now, let's talk about antibodies or immunoglobulins. There's some very important 4- or 5-star details out there that you really need to know for your exam. Let's start with the structure of antibodies. There are 2 heavy chain regions and 2 smaller light chain regions. And at the very tip of each of these is a hypervariable region. It's on the amino-terminal portion of each of them. One important thing to know is that the antibody is held together by multiple disulfide bonds. Anytime you want to hold a polypeptide together outside the cell, you need a very strong type of bond, like a disulfide bond. Disulfide bonds are some of the strongest bonds around, so just remember that as a generalization. Disulfide bonds keep things together outside of the cell. And it's particularly important to know where these disulfide bonds are found on the antibody. Again, these picky details are unfortunately important. There are disulfide bonds within the heavy chains and the light chains themselves. There are disulfide bonds connecting the heavy chain to the light chain, and there are disulfide bonds connecting 2 heavy chains to one another. So these disulfide bonds are very important to maintaining the structure and function of antibodies. Now, you can talk about antibodies based on their structure, which involves the heavy chain and the light chain. Or you can look at antibodies based on their function. Antibodies have 2 different functional components, the Fab region and the Fc region. There's a Fab region, or antigen binding fragment, which includes that variable region on the amino-terminal end of the antibody. AB for antigen binding. This is what determines the idiotype, the unique antigen to which that antibody can bind. This antigen binding pocket can only bind to one very specific antigen. Again, this is a part of the adaptive immune system, which is very specific. And just like the T cell receptors can only bind to a very specific antigen, it's the same is true for antibodies. Plasma cells will express only one particular type of antibody that binds to one particular antigen. And that Fab fragment consists of 2 light chain regions and 2 heavy chain regions. You need all 4 regions to make the Fab fragment. Then, the Fc region is the constant region, found on the carboxy terminal of the antibody. It only includes the heavy chains, and the Fc region is the part of the antibody that can bind to the complement. So there are lots of C's associated with the Fc region. It's constant, it's on the carboxy terminal, and its complement binding. We also saw earlier that this portion of the antibody is recognized by CD16 on NK cells. And it's the Fc portion that determines the isotype of that antibody, whether it's an IgG antibody, or IgM, or IgA, or whatever. We'll talk about the function of those specific isotypes in a little bit. So the Fab portion determines the idiotype, and the Fc portion determines the isotype. There are 5 different types of heavy chains: mu, delta, gamma, alpha, and epsilon. And each of these heavy chains corresponds to a specific antibody isotype. If an antibody has a mu chain, and that makes it an IgM antibody. Delta corresponds to IgD, gamma corresponds to IgG, alpha corresponds to IgA, and epsilon corresponding to IgE. Now, there are only 2 different types of light chains, 5 heavy chain regions and 2 types of light chains, lambda and kappa. There's no functional difference between lambda and kappa chains, not in the same way that there's functional difference between heavy chain isotypes. So an IgM antibody with a lambda light chain is really not that much difference from an IgM with a kappa light chain. But in humans, the normal ratio of kappa to lambda is about 2 to 1. For every 2 immunoglobulins with a kappa light chain you have, 1 immunoglobulin has a lambda light chain. So if you're looking at 150 different types of antibodies in a particular human, the odds are 100 of those antibodies will be kappa and 50 will be lambda. Now, how is that useful? Well, consider a case of multiple myeloma, which is a neoplasm of plasma cells, where one plasma cell undergoes malignant transformation and starts replicating itself over and over and over. In multiple myeloma, you have a huge proliferation of an original malignant plasma cell. So all of those malignant cells are producing the exact same antibody with the same Fab fragment and the same light chains. And that means the ratio of lambda and kappa is going to be skewed. Instead of seeing 2 to 1, it might be 50 to 1 or 1 to 50. And then one last detail regarding the structure of antibodies is the terminology used to describe the composition of the antibody in terms of light chains and heavy chains. So, in an IgG antibody, there are 2 gamma regions, which would be your heavy chains. And it would either have to be 2 lambda or 2 kappa light chains, right? So, IgG would have 2 gamma and 2 lambda, or 2 gamma, 2 kappa. So, what do antibodies do? The short answer is that they bind antigens, either soluble antigens that are just floating around in the serum, or they bind to surface antigens on the pathogens, such as antigens on the surfaces of bacteria. This can neutralize the pathogen, making it harder for the bacteria to cause trouble. This can opsonize the pathogen, making it tastier to the macrophages to gobble it up. And antibodies can activate complement, which eventually build membrane attack complexes and kill the bacteria. We'll talk about that in the video on complement. Let's think for just a moment about how the hypervariable Fab region of antibodies are created on a genetic level. DNA is not supposed to vary, right? But with antibodies, we need a way to create variations in the genetic code for the Fab region in order to create antibodies that can bind every possible antigen you can imagine. The process that allows this genetic variability is called VDJ recombination. The VDJ recombination is rearrangements of the specific segments of DNA, which are called the variable V segment, the diversity D segment, and the joining J segment. These segments compose the coding regions for each specific antigen receptor on B and T cells. So this process explains not only antibody variability, but also the variability in antigen specificity we see with T cell receptors. Any protein on a B cell or T cell that's binding to an antigen is going to be created from this VDJ recombination process. The rearrangement process begins at the breaks in the double-stranded DNA at the recombination signal sequences, which is abbreviated RSS. These signal sequences flank each of the V, D, and J coding regions. After the DNA has broken at these specific breakpoints, the VDJ recombination is initiated by a protein called the recombination activating gene complex, or RAG, RAG1 and RAG2. RAG1 and RAG2 recognize the RSS sequences that flank each of these coding regions. That's going to initiate the recombination process. And then one last important point here is the mutation in either of these RAG genes, and it causes the inability to initiate this VDJ rearrangement and the arrest of a B cell and T cell development. This recombination is fundamental for the adaptive immune system to work. If a B cell can't generate immunoglobulins, it's pretty worthless. So make sure you know the big steps in the recombination process. Now, let's talk about the different immunoglobulin isotypes. We said that IgM and IgD are found on the surface of B cells, but some isotypes are found in the circulating blood. Let's talk about those circulating immunoglobulins first. The main circulating immunoglobulin is IgG. It is the most abundant immunoglobulin in the blood, and the IgG is part of the delayed immune response to an antigen. When you encounter an antigen for the first time, you don't have any plasma cells primed to make the antibodies against that antigen. It takes weeks to develop a population of B cells that make that specific antibody idiotype and convert them to plasma cells and start cranking out that antibody. That's why IgG is so important in the secondary immune response, or delayed immune response. Let's talk about the important functional characteristics of IgG. IgG fixes complement. It crosses the placenta. So this is the immunoglobulin that's going to keep your baby healthy, not only in utero, but also for the several weeks after the birth. IgG opsonizes bacteria and makes them more tasty for macrophages. And IgG binds to the viruses and bacteria toxins and neutralizes them. So basically, IgG can do all of those things we mentioned earlier as the function of antibodies. IgG is sort of your prototypical antibody. Let's move on to IgA antibody. IgA antibody is the primary antibody that's secreted by MALT, the mucosa-associated lymphoid tissue. IgA secreting plasma cells are found in the mucous membrane and along the GI tract. IgA exists as a monomer in circulation, but the secretory IgA, which is secreted by the GI tract in tears and mucus, saliva, breast milk, or colostrum. IgA is a dimer with the constant regions linked end to end, so the Fab fragments are pointing in two different directions. In order to get into the lumen of the gut, secretory IgA has to pass through the epithelial cells by a process known as transcytosis. And while it's passing through, it picks up glycopeptide called secretory that wraps around the IgA, kind of like a suit of armor to protect it from the gastric acid or whatever harsh environment it's heading into. Next, there's IgE, which mediates immunity to parasites, especially parasitic worm infections. It does this by activating eosinophils. You can remember IgE and eosinophils. IgE's other claim to fame is that it can bind to a specific receptor found on mast cells and basophils, and then when that bound IgE binds to an antigen, it starts cross-linking with other IgE antibodies. And that causes the mast cells and basophils to degranulate and start releasing histamine and a bunch of other substances that can lead to type 1 hypersensitivity reactions, or allergic reactions. So for IgE, think about parasites and think about allergies. So those three, IgG, IgA, and IgE, are all the circulating antibodies. And we said that IgM and IgD are surface antibodies. I want to talk about IgM next because it swings both ways. It's found on the surface of B cells and it's also found in circulation. When it's attached to the surface of B cells, IgM is a monomer, but in circulation, it exists as a pentamer with 5 IgM antibodies linked together, kind of like spokes on a wheel. IgM is a part of the primary immune response of an antigen. When you encounter an antigen for the first time, the antibodies generated are mostly IgM. IgM is the primary immune response, and IgG is the secondary immune response. So, Say a patient comes in with an encephalitis-like illness and you're worried he has West Nile virus. You're going to check the patient's blood for both IgM and IgG antibodies to West Nile. If the anti-West Nile virus IgM antibody is elevated, it means the patient does in fact have an acute infection. If the IgG antibody is elevated, that means the patient had West Nile virus sometime in the past, but it's not necessarily an acute infection. The other thing you should know about IgM, that it's different from IgG, is that IgM does not cross the placenta, mainly because that pentamer is just so big, it can't. And then finally we have IgD. We don't really know too much about IgD, except that it's found on the surface of B cells, and there are also very low levels of IgD found in the serum. That's it for antibodies. So now let's work through the end of session quiz. Let's go over the answers. First question, which surface markers are found on B cells? The ones that are unique to B cells include CD19, CD20, CD21, IgM, and IgD. Then they also have MHC1 because all nucleated cells express MHC1, and they have MHC2 and a B7 protein, either CD80 or 86, and also CD40. Which immunoglobulin isotype fits each of the following statements? The first one, associated with allergies, binds to mast cells and basophils, and causes them to release histamine. That's IgE. The most abundant immunoglobulin in the blood. IgG. IgA is more abundant overall, but IgG is the most abundant immunoglobulin in the blood. Present in large quantities on the surface of many B cells is both IgD and IgM. Crosses the placenta and additionally confers immunity to the neonates. That's IgG. Can occur as a dimer. Secretory IgA. Largely confined to the intravascular pool and is predominant early antibody frequently seen in response to infectious organism with complex antigens. This is IgM. Distributed evenly between the intravascular and extravascular pools is IgG. Predominant immunoglobulin in secretions, that's IgA. And can be a pentamer, that's IgM. Last one. What initiates recombination of VDJ sequences? That's the RAG1 and RAG2 complexes. Remember, RAG stands for Recombination Activating Gene. That's it for this video. We'll see you next time.